Hi, I am Shamresh Mondol, Senior Lecturer, Department of Biology, Milestone College, Uttara. How are you? I hope you are well. In the previous class, you learned about the asexual reproduction and parthenogenesis. Today's our topic is artificial reproduction or artificial hybridization process. From this class, we can learn about what is artificial reproduction, what is emasculation, the process of artificial hybridization or reproduction process, and at last the significance of artificial reproduction. So let's start our today's class. In this process, at first we have to learn about what is artificial reproduction or what is artificial hybridization. Artificial hybridization is the process of crossing two genetically different individuals to create new genotypes of high-yielding varieties of plant. So, in this process, we can get a new high-yielding variety. Method of artificial hybridization. This artificial hybridization process occurred step by step. The first step of artificial hybridization process is to select parents. That means parent selection. Second step is the artificial selfing of parents. And third step, emasculation of female parents. Fourth step, bagging. Fifth step, artificial crossing. Sixth step, labeling. And step number 7, collection and preservation of seeds. And last of all, step number 8, raising of F generation. F means filial. So to complete this artificial reproduction, we have to follow this 8 steps. First step of this artificial hybridization process is parent selection. The first step of artificial hybridization is to select a healthy male and female parents that should be participate in the process. Second point, artificial selfing of parents. Artificial selfing of parents. If the plant is cross fertilizing in nature, then it would be artificially selfing. As a result, no possibilities of mixture of unexpected traits and plant confirm its homozygosy. So the main notion of this artificial selfing of parents to confirm its homozygosy. This is a picture of pollination process. We know that pollination are two types, cross pollination and self pollination. Cross pollination occurred in between the two flowers of two different plants, and self pollination occurred in between same plant's flower. Self pollination are two types. First one is autogamy. Autogamy pollination occurred in between the same plant and same flower. That means autogamy self pollination occurred in same flower of same plants. And another types of self pollination is getonogamy and getonogamy. Getonogamy, the self pollination which is occurred in between the two flower of same plant is known as getonogamy. Third step. Third step is known as emasculation of female plants or female parents. Removal of stamen or anther or killing the pollen of flower without the female reproductive organ, known as emasculation. In bisexual flower, emasculation is essential to prevent of cell pollination. So the main reason of emasculation is the protection of cell pollination or to protect the cell pollinating process. This is a picture of a emasculation process. This flower is a bisexual flower. From this flower, the male sexual organ removed. Fourth step is bagging. Immediately after emasculation process, the flower or inflorescences enclosed with suitable bags of appropriate size to prevent random cross pollination. So the main reason of bagging step to prevent random cross pollination. This is a picture of bagging step. Here a emasculate flower covered by a bag. Step number 5. Artificial crossing. The pollen grain collected from a desired male parent should be transferred to a emasculate flower with a camel hair brush. This is normally done in the morning hours during the anthesis. That means the time of flower blooming. The flowers are bagged immediately after artificial crossing. 6th step, labeling. The next step is to label seed parents. Each pollinated flower should bear a label containing the name of the seed parents, the letter X, the name of the pollen parents, and the date of crossing. Step number 7. Name, collection and preservation of seeds. When the seeds are completely developed, this should be harvested as soon as possible. And then seed stored in a cool and dry place for future sowing and cell pollination. So in this step, we have to collect developed seed after harvesting and preserve it in a cool and dry place for future sowing and cell pollination. 8th step and last step, raising of F generation. F means filial. The seed produced by first hybridization if shown in the field. Next year, the plant which are produces by intercrossing of F1 generation or F1 hybrids resulting F2 generation or second filial, and in the same way F6 generation produces and selected as a this F6 generation selected as a new variety. And this is all about the process of artificial hybridization process. Now we have to learn about the significance of artificial reproduction. By the process of artificial reproduction or artificial hybridization, we produce high yielding varieties of different types of crops such as rice. B, that means Bangladesh Rice Research Institute developed diabetic rice, vitamin A and zinc enriched rice. Also produced many types of high yielding variety, flood resistance, salinity tolerance variety. Also they produced BR39, BR40, BR28, BR10. This BR10 also known as Mukta, BR14, BR14 also known as Gazi. These are the different types of high yielding variety of rice. High yielding variety of wheat. Norman Ernest Borlaug has developed semi-drop high yielding disease resistance wheat. He was familiar as the father of green revolution. Some high-yielding variety of wheat in Bangladesh are Shatabdi, Pradeep, Bijoy, Bari, Barigom 25 and Barigom 26. These are the name of the different types of high-yielding variety of wheat. Next corn is maize. Maize hybrids were first commercially grown in the 1930s. The notable hybrid varieties of maize are KS 9451, Pusha 11, Pusha Hybrid 1, Pusha Hybrid 2. These are the different types of high yielding variety of maize. By the process of hybridization, many types of high yielding varieties of fruits produced, such as mango, such as lychee, banana, orange. Third point, high yielding varieties of vegetable like cabbage, broccoli, cucumber, sweet gourd, etc. These are the different types of high yielding varieties of vegetable. Fourth point, flower and orchid. Flower like rose, gladiolus, tuberose, etc. Most of the orchid are produced through the hybridization process. Next point, and insect resistance variety. Some hybridization technique focus on combining strong virus, bacteria, fungi and insect resistance characteristics from parents plants. 7 point, uniform plants. F1 hybrids produce uniform plants. Uniform growth selection often results in earlier maturity. Next point, seedless fruit. Seedless fruit produced by the process of artificial hybridization process, such as seedless banana, seedless grapes, seedless watermelon, seedless lemon, seedless apple are developed through the artificial propagation. This is all about the significance of artificial hybridization process or artificial reproduction. So I think you can easily describe the process of artificial reproduction or artificial hybridization process, and you can also write down the process. So next day homework, learn and write the process of artificial reproduction or hybridization. Write down the significance of artificial reproduction or hybridization. If you fail to understand anything, please contact your subject teacher. Thank you everybody.